Hi, I am Priyanka and I am a cancer biology student. Today I am going to do a presentation on the topic Design and Validation of BRCA1 Expression Vector in MC7 MCF7 cell line. For this, I use the Homo sapien gene that is BRCA1 which encodes a nuclear phosphoprotein that is BRCA1 protein that plays a role in maintaining genomic stability and it also acts as a tumor suppressor. And here I use the expression vector TCMV6 entry vector. My aim is to design a BCRCA1 expression vector and validate it in MCF7 cell line. The first step is that insertion of this BRCA1 in PCMV6 enriched expression vector. For this, first I need the cDNA of BRCA1 and using this cDNA, I need to design a primer. I designed the primer and with the help of this primer, we need to amplify in PCR, amplification. And after amplification, we need to digest the PCR product and the PCMV6 vector using restriction enzyme. And then purify the digested BRCA1 fragment and the linearized PCMV6 vector from a agarose gel to remove the excess primers, enzymes, and nucleotides. And then ligase the BRCA1 insert into the linearized PCMV6 entry vector to form a circular plasmid with a BRCA1 insert. After ligation, we get a circular plasmid and we need to transfer it to a bacterial cell. Here I use E. coli, which is the competent cell, and I done all these steps and heat shock it and I spread it in on agar plate and kept it for incubation for overnight to grow colonies. After overnight incubation, um, and I obtain the colonies with the ligated BCRCA1. I need to extract the plasmid from this. For this, I done series of centrifugation and I get the plasmid extract. And then we need to verify the sequence. For this, first we need to uh, restrict restriction digestion of the isolated DNA using the restriction enzyme and analyze the digested fragments on an agarose electrophoresis gel, confirming that the digestion results match the expected pattern, indicating the proper insertion of the BRCA1 gene. And then we need to sequence the plasmid DNA to verify the accuracy of the cloned sequence. Here I use the Sanger sequencing method for sequencing. Then we are moving to the last step that is transfecting this BRCA1 expression vector into our MCF7 cell line. MCF cell line is a uh, human breast cancer cells. For this, first I need to harvest the cell, detach the MCF7 cells from the culture flask and using trypsin EDTA, and then seed the desired amount of number of cells in each well for of the culture plate. And then the next step is to prepare a transfecting complex with the mixture, mix the BRCA1 expression vector with the transfection reagent and incubate this transfection vector complex for about 15 to 30 minutes at room temperature to allow the formation of the transfection complex. After forming the complexes, we just transfer it to the cell. That is the cell line MCF7. And we need to study that the gene is expressing or not. Here I use two technologies to validate the expression of the vector. Here first I use Western blotting and I also use immunofluorescence analysis and I successfully got this result. This is all about my study and these are my references which help in this study. And this is all about my project, my presentation, and thank you for your time.